Hello, everyone. Welcome back to the next edition of our general biology lectures. Today we're going to be getting into talking about the organization of a cell, the structure of a cell, and how we came to know these things about cells. We're going to start by talking a little bit of history, but then we'll get into identifying the different characteristics of cells and compare them versus viruses and how viruses don't have all of those characteristics, which is why we don't consider them life. We're going to compare the structural differences between the prokaryotic and the eukaryotic cells, as well as between the different eukaryotic cells of animals and plants and fungi. And then we'll get into talking about those individual components, those organelles within the eukaryotic cells, and the lack of organelles in the prokaryotic cells. And we're actually going to do that last part altogether in a bit of a activity. So that's a little summary of our sections. So first, let's talk about the sort of history of the cell. So cells now are pretty widely recognized as the smallest unit of life, at which that life that can function independently. The pieces of a cell can be separated, but they don't really function the same when they're separated from a cell. And then all life needs cells in order to do its functions. But cells we didn't really know in Western science. We did not know cells existed, really. We didn't know that life was made up of smaller units like this. It was until the mid 1600s when specifically Antony von Leeuwenhoek, not sure I'm saying that name right, but he's a Dutch scientist generally recognized as the father of microbiology as a field because he used a very simple microscope that he designed himself to discover, like, oh, hey, there are these little tiny critters, these little, we call them animalcules or something like that, where they're like little animal molecules that are floating around. They didn't really have the molecules as a defined thing yet either, but he saw that there were these unicellular and multicellular organisms that were not visible to the naked eye. And it was only through the assistance of a microscope that you can see things at that scale, see that level of detail. And that's where Robert Hooke comes into the story too, because around the same time he was also he was an Englishman who was designing all sorts of scientific equipment, was also a meteorologist and sort of a bit of a polymath, but he was critical to the development of some of the earliest compound microscopes like we use today. But that microscope technology was very simple back then. They used actual physical flame lamps that the light had to be concentrated into a point in order to illuminate microscopes. So that's how like the first light microscope came into being. But then fast forward a few hundred years and we start to have developed this technology to something a little bit more like the compound light microscopes that we see today, where you have the multiple objective lenses you can rotate into place. And then, of course, there have been further technological innovations. The first electron microscope was developed in 1933, or I think this is the first one, if you can correct me on that, but around earlier 1900s, mid 20th century, you start to see a lot more advancement in electron microscopy, which really allows you to see not just the cells and the rough structures, but you can really get a bigger picture of the detail of these different, not just the cells, but the macromolecules that they're made up of. And then around the mid 1800s, and this was developed into the 1900s, we have what we refer to as the unified cell theory. So all these discoveries of cells and how critical they were to life led scientists, specifically Matthias Schleiden and Theodor Schwann, to develop this into a unified theory, something that can be generally applied and has been repeatedly tested and supported across a wide variety of circumstances. Remember, that's how we think of a theory. So there are three main points to this to consider. First is that all organisms are made of one or more cells. Like that is what defines an organism, is that they're made up of a cell or cells, unicellular, multicellular organisms. And that is what makes cells the base unit of life. Anything smaller than a cell is not life, and everything above anything that is life has to be made up of cells, going back to the first point. And then what goes along with this, knowing that life is also something that reproduces itself, is those cells have to come from somewhere, and that what is true is they have to come from existing cells, whether it's through sexual reproduction or asexual reproduction, budding, and such. Those cells have to arise from previous cells. And then later on, there were more points added to this. So there are a lot more points in this unified cell theory, but those are the main three that were originally postulated and have stuck with it over the decades, the centuries at this point. So let's switch now to talking about what makes the components of a cell, what defines a cell, and what are its characteristics. The first of which is that cells are small. With some exceptions, there are some, you can think of a large ostrich egg is essentially one massive cell. But the reason why cells are small has to do with the surface area and their volume. So the thing is, just based on geometry, when you have a smaller object, it has a higher surface area to volume ratio. So you get more surface for it to interact with its surroundings versus a larger object. Both your surface area and your volume will increase, but your volume will increase a lot faster. So in this example with the cubes, you double each of the dimensions, you get 4 times the surface area, but you get 8 times the volume. So as you get larger, you get a lot more volume, a lot less surface area. So the things that are within that volume can't interact with the surroundings. So the things within a cell can't do as much interacting with the surroundings. Typically, cells end up being in this micrometer to hundreds of micrometers in diameter, with some exceptions, like I said, like large eggs are some of the largest cells in the living world, and so they can be pretty big. But generally, this function of it is what limits the structure. In order for it to function, it has to be relatively small. And then there are 5 main things that we think of across all cells. So all cells have a plasma membrane, which we've talked about lipids last week. These plasma membranes are made up of some layer of lipid, like phospholipids, that separate the cell's insides from its outside. So you've got this soft, flexible layer that separates it from the surroundings. Within that, you have your cytoplasm, which is kind of it's jelly-like because it has a whole bunch of things dissolved in it and floating around in it. It has a viscosity to it, but it's generally water, mostly water. And this cytoplasm, this jelly within the cell, forms the volume of the cell. It's where all the important bits are located. It's where the little molecules are floating around in. So you have to have the outer layer of the plasma membrane, then you have to have a cytoplasm. You have to be filled with something. And 2 things that it has to be filled with are DNA, ribosome, and ribosomes. So DNA is that genetic material that provides information on the production of proteins, the proteins that are to be made, and then those proteins can then do work. Then ribosomes are made up of other genetic material. They're made up of RNA as well as proteins. and ribosomes are what are involved in synthesizing the proteins based off of DNA and RNA. So we have our basic pieces now. We have our plasma membrane, our cell membrane, and we have our cytoplasm, the stuff filling it up. And then we have some DNA in there somewhere. We have some RNA, including in the form of ribosomes, which are structures. We consider ribosomes, we sometimes refer to ribosomes as organelles, but they're not membrane-bound organelles, which is why prokaryotes have these as well. So eukaryotes, prokaryotes all have these. The one thing that sort of going without saying there is all cells have proteins as well. But then we can compare that to viruses and what makes them different. So a virus does have genetic information, so it has DNA or RNA that are involved in its reproduction. It often has a protein coat, a capsid, that surrounds that genetic material and whatever else is filling it. It has proteins, it has enzymes that are involved in doing the work for it to infect or to infect living things, because viruses are not considered living. And then they sometimes have an envelope of lipids around them. Sometimes they steal that from surrounding from actual cells. So they don't produce these themselves usually. But really, all they have is they have DNA, they have proteins. They have the genetic material, they don't really have a cytoplasm, they don't really have any membrane-bound organelles, they don't even have their own plasma membrane usually. So there are a lot of different types of viruses and how they operate, how they go about doing the things that they do and infecting other cells. Here we're going to watch a quick video talking about HIV, the human immunodeficiency virus, and how it operates as a retrovirus, where it sort of hijacks retroviruses sort of hijack the genetic operations of other cells, of human cells in this case, to make them to reproduce themselves. They're one of these ones that even steals some of the membrane that we're talking about. So we'll watch this here. Make this a little larger.
This is HIV. It's a typical retrovirus, meaning that it has an outer envelope, and in the center it has 2 copies of RNA, as well as an enzyme here in blue that's reverse transcriptase, which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein. That causes a conformational change and allows a second receptor to grab hold of the envelope. This is the chemokine co-receptor. It's also called CCR5, and we'll talk about that more. What happens now is that the stalk of the envelope protein pierces through from the virus into the host cell and starts to draw the cell membrane and the viral membrane together. And what ultimately happens is fusion of those 2 membranes, and the viral genetic material is injected essentially into the cell, and the envelope protein is left at the cell surface. The virus has a matrix and a capsid protein, shown here in green and red, that essentially are digested when it enters into the cell. That releases viral enzymes and the viral RNA. And here we have reverse transcriptase, which takes the viral RNA and, using host nucleotides, converts that viral RNA into a single strand of DNA. While it does that, it makes some random errors, which is characteristic of reverse transcriptase. It has very poor proofreading activity. That single-stranded DNA now is again reverse transcribed into a double-stranded DNA. At that point, another enzyme that has come in with the virus in the beginning, called integrase, essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and it basically, the integrase enzyme, makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum. And it's shuttled then through the endoplasmic reticulum and taken to the cell surface, where at the cell surface it becomes embedded in the cellular membrane, and at this point coalescing with other envelope proteins that have been produced. You have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced that allow for translation of other viral proteins. So here are additional viral proteins being made, which are going to be used to make up the key components that the virus ultimately is going to need. These are transported again to the cell surface, to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make up the final virion. And now you have a mature infectious virion that can go on now to infect other cells. Once that happens, now the cell can produce tons of viruses, and this is really what then keeps the whole process going.
Yeah, that was a little illustration of not just how viruses operate, especially retroviruses and their sort of nasty habit of hijacking cell processes, but this is sort of a good little introduction to some of that general flow of how things operate in a cell. So you have DNA getting transcribed into RNA, and then that RNA gets translated to form proteins, and then a virus like HIV does just sort of can't really do those things itself. It has its proteins, it has its envelope. It doesn't produce its own envelope. It steals that from the cell. It doesn't produce its own proteins. It just inserts its RNA, inserts its little code into our own DNA, which is permanently there, which is what makes for lifelong infection, like they said. But then that ends up getting treated by our own cells as a piece of our own DNA to produce something that isn't for our own cells' function. And yeah, so a little bit of an introduction into that process of transcription and translation in the ribosomes, and how viruses can't do it on their own. And oh shoot, I already have this selected here. If you have already gone ahead to the next slide, you may have already selected what you think are the things that viruses have in common with cells, which is that they have the proteins, they have the genetic material, but they don't have anything to do that stuff, to do the basic functions of life on their own. So they need the cells still to operate. They need those ribosomes, they need the plasma membranes, cytoplasm. One thing you may also notice is one thing they don't have in common is they're also a lot smaller than even the prokaryotic cells. If we go back a couple slides, to our, here we have some bacteria cells, then those little bacteriophage viruses are much smaller than the bacteria themselves. So, if we were in person, I would tell you to turn to your neighbor, but again, just check in on your notes. Make sure you understand what is important about microscopes, which is that we can't see our fundamental pieces of life without them. The 5 components of the cell, remember we had 4 of them listed pretty explicitly. We have the plasma membrane, the cytoplasm, the genetic material, DNA and RNA, and ribosomes, but then sort of inherent part of the plasma membrane and part of the ribosomes, you can't have a cell without proteins. So proteins are sort of that 5th component of a cell. And then acellular microbes, which are our viruses. They are still microorganisms almost, except the only things they share with us are the genetic material and proteins. They don't really have much else other than that, at least not of their own. All right, and now let's talk about our main domains of life and how their cells are different. So remember we have our prokaryotic organisms and we have our eukaryotic organisms. Prokaryotic cells are those in the domains of bacteria and archaea. They have, in addition to all the 5 main things, they have a few certain characteristics. They don't have a nucleus, but they do have a nucleoid region. There's a part of the cytoplasm in which the genetic material typically resides. They have a cell wall in addition to their plasma membrane, so they have a harder structure. It's normally made of some polysaccharides, and I think some other materials, not cellulose, that's in the plants, like we'll talk about. And then they normally have these attached to a lot of bacteria have a capsid, an additional sort of polysaccharide hard shell outside of the cell membrane and the cell wall. And then attached to the cell wall or to the capsid there are these external structures for movement. So those structures are often a long flagellum or long flagellae. Then we have these pili, these little smaller wiggly bits that help them move. So this is generally what they look like. They are our generalized picture of a prokaryotic cell. Remembering that all of these sorts of illustrations are general examples. Not every prokaryotic cell has this kind of shape or even has all of these structures, but we want to show them all in one little cartoon. Oh, and they do have ribosomes. They're not membrane-bound organelles, but they do have ribosomes, which are made up of RNA and proteins, and they help to synthesize proteins. You can't have the sort of synthesis of proteins that are needed to make up any cell without having ribosomes. And like I was saying, there's no membrane bounding that DNA. So you have just a general region in which the DNA hangs out. And yeah. That is in contrast with, of course, the eukaryotic cells. So these are the whole other domain of life to which we belong, as well as all the plants and animals and a lot of things that we vaguely call protists. But what makes us different is that we have membrane-bound nucleus and other membrane-bound organelles within us. Within our cells, that is. And also, these are much larger. So the bacteria are larger than the viruses, and the other acellular microbes. And then eukaryotic cells are much larger than prokaryotic cells in general. So you can maybe even see on this, some of those little dots right there might possibly be little bacteria contaminating these slides. Much smaller. And talking about the different types of eukaryotic cells, so within the eukaryotes, the different kingdoms have some general differences between what we have in our cells. Us animals, we don't have cell walls. We don't have a rigid structure on the outside of us, of our cells. And our cells take on a lot of different shapes. Like I was saying, we use these sort of general cartoons, like the one on the right, so we can represent cells that have a single shape and all the important bits in it. But then you have such a wide variety of cells. Here, what we're showing are an ovum, which has a bunch of accessory cells around it. So one large egg cell, a erythrocyte, a red blood cell, which is a very reduced version of a cell. Like it's actually missing a lot of the things that most of the rest of our cells have. Then we have much more complicated shapes like that of a neuron shown here at the bottom. Then we have muscle fibers. And again, remember, all these different forms have to do with their different functions that they have in our body. And getting into plant cells, so plant cells still, they also have a variety of forms and functions. I'm not going to show them all here, but plants have certain specialized organelles, the most important of which are the chloroplasts, which are the small little organelles in which photosynthesis occurs, which is critical to their operating as as autotrophs, especially critical to their function, taking the light and turning that into energy, using that to build sugars. And then plant cells also have a vacuole in them, which is usually just filled with fluid, filled with water. This is what sort of enables a plant to sort of regulate its sort of turgidity, its fullness, as you would say. So by having a bunch of water in this membrane-bound, just membrane sac within the cell, you can sort of push that cell membrane up against the cell wall and really get it full. If the water vacuole is completely empty and then there's a loss of fluid from the cell itself, the cell membrane can get sort of shrunk away from the cell wall. And what about the fungi, though? What do they have in common with and different from the animals and plants? Well, keep in mind that fungi are heterotrophs. They don't really have chloroplasts, but they do have cell walls, just as the plants do. So they don't, unlike the animals, fungi have cell walls. Cell walls of fungi are also made up of a different thing. With plants, they're made up of cellulose primarily. With fungi, they're primarily made up of chitin, which is a similar kind of molecule to what the exoskeletons of bugs are made up of arthropods are made up of chitin. But so you have different things, but plants and fungi both have cell walls, but the fungi won't really have the chloroplasts because they're not producing their own food through photosynthesis. So keep that in mind when we're drawing these all together. So real quick, we want to remind ourselves which of these is larger. Those acellular microbes, the viruses, are definitely the smallest, and then the eukaryotes are much bigger than the prokaryotes, so this is how we would rank them. Oh, I got it backwards. I misread this. Small, that's why you read the instructions. So should put the smallest at the top and the largest at the bottom. Anyway, so we want to think a little bit about this. So talked about the differences between the prokaryotic and the eukaryotic cells. We also want to think about what are the disadvantages to having these different types of cells. So what is potential advantages to being small versus large? I think this is a great thing to bring up in our discussion, and we can chat about it a bit more at length. So the last thing, that's all I have for this lecture. Well, almost the last thing I have for this lecture. What we're going to be doing next is we're going to let you teach, essentially. So I could sort of just rattle off the different types of organelles and what they are, what sort of function they have. But what's more helpful here is for you to look it up yourself. So what we'll do is we'll talk about this more when we meet. We'll come up with a plan for this, but we have a shared Google Slide presentation where each two of you will get a different organelle, and you will get to fill in some of that information yourself, and then we'll sort of come together and talk about it as a group, as a class. And the last thing I have for you today is I wanted to share with this video called Inner Life of the Cell, which was produced a while ago at this point, but it's this really fascinating animation, very soothing, has some nice calming dramatic music to go along with it. But this is a computer simulation of each of just what's going on in your typical cell, how different parts are sort of moving about. And what's interesting is each of these are fully accurate to the way things are within some of our cells. Specifically, I think we're looking at a leukocyte or a white blood cell, or maybe it's some other type of white blood cell. If you want a little bit of an explanation of what's going on, there is this helpful video that was done where they pause and actually talk about the different pieces. I couldn't explain everything that's going on here off the top of my head, but yeah, with that, I'll just leave you with this video, put it on, and be quiet, and I will see you in class. All right, y'all. Thanks for watching. See you in class.